Good afternoon, everyone. Thank you for joining us on our first demo day ever. In order to share with you the results we have achieved after an intense 18-month-long work cycle, we decided it was pertinent to invite you to participate in this gathering where we can take the time to review our ideation, development, and implementation processes in detail. Since this is the first time we're carrying out this type of presentation, we'll start by explaining the main reasons for the existence of STEM and what motivates us to come to work every day. We also want to take this opportunity to address how challenging 2020 has been. We hope all of you stay safe and stay healthy during what remains of this pandemic. 2020 has been a special year for humanity. It forced us to confront the darkest sides of our reality and made us stand humble before the fragility of it. As an organization, we feel urged to contribute with new instruments to be able to face these unprecedented challenges. But something else happened in 2020 that a lot of people don't know about, because it was overshadowed by the pandemic and by other major events we faced last year. In this graph, we can see how the volume of living biomass changed throughout the years, from our first agricultural revolution until today. This brings to our attention the impacts of humanity's advance on nature, since it shows that the amount of living biomass cut in half throughout the last 12,000 years. This graph gets even scarier when we include the volume of anthropogenic mass on the same timeline. We can see that in the 1900s, the amount of anthropogenic mass was equivalent to 3% of the world's living biomass. And this percentage already indicated that the production of living biomass globally had fallen 50%. Throughout the last 100 years, the anthropogenic mass increase rate skyrocketed to unprecedented levels. What makes 2020 a unique year in the history of humanity is that the total volume of anthropogenic mass matched the total amount of biomass globally, which means it matched the total volume of everything that is alive on the planet. This graph provides a striking perspective on the rate of change in the relationship between humanity and nature. It illustrates the absolute limits of the systems that we live in and the preposterous uncertainty that our future holds. Where do we go from here? Last year, a 30 gigaton growth in anthropogenic mass was registered. We can only hope that this number keeps increasing year after year, since it's key to solve humanity's systemic problems. This number is forecasted to reach 3 teratons by 2040, almost 3 times the current world biomass. On the other hand, the biosphere will not be able to endure this trend in a zero-sum logic. We have to make sure that conquering the great challenges of humanity doesn't extinguish the planet's capability of sustaining life. Solving how we obtain the materials we need in a sustainable way is one of the great challenges of humanity. Transforming our relationship with nature in order to restore and sustain the planet's biomass productivity is indivisible with the prosperity of humanity. When we try to measure the scale of this challenge, we see 2 large groups of materials. First one is agricultural, biomass, animal population, mass, and plastic waste, with a total volume of 25 gigatons. And the other one is the requirement of inanimated and inorganic mass for 35 gigatons per year. So we ask the question, is it possible to obtain all the mass that humanity needs without resorting to extractive exploitation and the contamination of our ecosystems? We estimate a biomass production platform that can supply 50 gigatons per year to be able to replace the extractive exploitation of our ecosystems. Today's prevalent global biomass production methods are extremely inefficient when we look at their water, carbon, and land footprint. This inefficiency becomes evident if we compare the total decline in biomass worldwide since the beginning of the first agricultural revolution by 1200 gigatons versus what we gained from the global biomass production of agriculture for a total of 11 gigatons, 100 times less. Even if we were willing to lose this biomass production on a systemic level, we would need to almost double the extension of arable land globally in order to achieve a production of 50 gigatons. Not to mention the challenge of genetic engineering of crops subject to climate variability and the deepening of the exploitation and extraction of our ecosystems. If we look for these 50 gigatons per year as building blocks for the production of biomass, we see two great sources that could be connected to this new supply chain. The first is the CO2 emissions that rise to 36 gigatons per year. Curiously, a volume very similar to the annual inanimate anthropogenic mass increase, making these CO2 emissions the main fuel for the production of humanity's infrastructures brings the opportunity to be able to immobilize those carbon dioxide banks for long periods of time and dissociate them from the atmosphere. Secondly, there is a sleeping giant capable of producing an immense biomass bank, our ecosystems. Allowing our ecosystems to recover their basal biomass productivity is one of the greatest opportunities we have to transition to a scenario of biomass abundance and establish a sustainable relationship with nature. Just from this initiative, we could have 24 times greater biomass production that we need. If this were the case, then the volume of molecular fuel for the production of bio-based materials available would be more than enough to fuel the growth of humanity, even in the more ambitious projections. However, abundance of biomass doesn't mean abundance of bio-based materials, and here is where bioprocesses have a transformative role. A bioprocess is a specific process that uses living things to transform matter into desired products. In this technology lies the solution to automate the transformation of molecular fuel into bio-based materials, tailor-made and customized, considering the rational availability of resources. The prosperity of nature is the fuel for the prosperity of humanity. Following this logic, the next step is to figure out how we can take advantage of this abundance of biomass through the use of bioprocesses. When we study the efficiency of technologies for biomass production, we find that bioprocesses have an efficiency between 1 and 2 orders of magnitude higher than other biomass production technologies, which positions bioprocesses as one of the most promising technologies to meet the requirement for bio-based material production. This technology even allows us to expand the use of inorganic materials available in the atmosphere, such as nitrogen. Today, worldwide, there is an installed capacity to process 250 tons of biomass per year. And we produce around 110 million tons of biomass, bio-based materials per year. Surprisingly, 95% of it is used for the production of bioethanol and alcoholic beverages. Even though the global carbohydrate supply chain still depends on domesticated crops, this technology allows the use of various carbohydrate sources that are compatible with the sustainable harvest of biomass from ecosystems without disturbing them, and even funneling the CO2 emissions as building blocks for the production of biomass. We also predict a significant redistribution in the use of bioprocesses. We expect the production of therapeutics, nutraceutics, and bio-based materials to take a more protagonistic role in the future of the industry in the next decades. In order to recover our ecosystems and develop bioprocesses that allow us to use alternative sources of carbohydrate to produce the amount of living biomass and inanimate mass that humanity needs, we need to increase the installed capacity to produce biomass by 450 times. We are convinced that we still have the opportunity to reach this goal in time, even though the bioprocessing industry will look very different from what it looks today, since more than 99% of the industry has yet to be built. Now, what have we been not able to achieve so far with this 0.23% of the industry? Until today, more than 100 years after the implementation of large-scale industrial bioprocesses, This technology continues to exist in a way that is completely detached from the daily experience of society and confined to very few spaces on the planet where you can actually make the most out of the benefits of this technology, using it to design as a design tool in order to solve real problems. We need a technology platform and a design language that expands the number of people thinking about bioprocesses as a tool to solve problems and offer products and services locally and globally. The decentralization and democratization of biotechnology is the only way in which we will be able to transition from value chains that are intrinsically oil-based or oil-centered to value chains that are intrinsically biomass-based or biomass-centered. We see three large areas of work to take into account in this regard. The first one is replicability. The bioprocess as a software. Develop tools that are able to deliver the same results for the target bioprocess regardless of the geolocation and scale of production. The second one is scalability. Once we are able to design a successful bioprocesses, we can access immediately to the infrastructure needed to scale it accordingly immediately. And the third one is reliability. Have an efficient and competitive logistic integration facing alternative materials. On the other hand, we see a great challenge in building a community of experts that can catalyze and effectively operate the growth of this industry. The new value chain of biotech industry 4.0 is already becoming a reality, where an interdisciplinary team can carry out an entire process of research, development, approval, production, and distribution of a biotech product from the cloud. For this, it's key to translate and simplify the language of biomanufacturing, as it has been done for coding. In the context of this conversation, we see that the industry makes a very intensive use of plastic and that it generates non-degradable and dangerous waste. Incorporating a design that contemplates circular economies is a great opportunity for the advancement of this technology to be synergistic with the role that it comes to fulfill. On the other hand, this 0.23% of the industry has been able to achieve many things. We understand that the biomanufacturing industry, in particular biopharma, is a fundamental industrial complex to supply the therapeutic needs of society and contain threats such as pandemics, as well as producing new treatments for orphan pathologies. This industry has allowed us to reach an unprecedented record time response regarding the development, testing, production, and distribution of vaccines and treatments against COVID. When we analyze the history of this industry complex, we see that since 2008, 40 antibody products have been approved by the FDA. However, last year alone, 18 new molecules were given the green light to be commercialized. In addition, 86 new antibodies are currently undergoing clinical trials. By 2025, this market is going to double from $150 billion to $300 billion of market share. And there are approximately 500 approved antibodies on the horizon for 2030. The global antibody production demand is projected to double over the next 5 years. And this is an industry that it took 45 years to be established and is facing unprecedented growth, and biomanufacturing will become the biggest bottleneck in the value chain. If we now look at the cell therapy industry, we can see that it also shares this behavior and is even more accelerated, given that it's leveraging an existing biopharma infrastructure. In 2017, the FDA approved the first cell therapy. In 2020, it had already approved 7, and it is predicted that this year at least 13 new cell treatments will be approved. By 2025, it is expected that more than 80 approved treatments will be commercialized, which means that in 8 years, the cell therapy industry would have reached the same number of treatments that took the antibody industry 45 years to approve. In the horizon for 2030, we can expect approximately 300 treatments approved. And to expand the context of the volume of work that is currently being carried out in this area, there are more than 800 cell treatments submitted in the process of evaluation. When we take into account what this represents from a manufacturing point of view and the current installed capacity available, we predict a growth of at least 10x by 2030. Access to certified facilities to get quality and scale by manufacturing represent an extremely scarce asset. Big Pharma is fighting to acquire this and secure their supply chain, given this forecasted scenario. Biomanufacturing technology is at the center of this problem, largely explaining this scarcity because of the difficulty to scale and replicate. 2020 made clear how essential it is to have resilient, streamlined qualification processes and supply chains for the biopharma industry around the world. The increased demand for manufacturing of COVID antibody treatments and other biologics addressing COVID-related syndromes just puts even more strain on this system. At STEM, we believe that to close this challenging gap, we need bold solutions so that the biomanufacturing industry can be the bridge between the prosperity of nature and the prosperity of humanity. So we set out to reinvent the way in which we implement bioprocesses, incorporating state-of-the-art technologies that are compatible with the role that we envision for them. Setting the goal of designing a plug-and-play machine that can carry out continuous bioprocesses automatically and implement a cloud-supported program which has replicability insensitive to scale, which can be massively built, where the user community can develop protocols of production and scale their processes by renting available capacity in the network. We call this machine the bioprocessor. To develop a tool that gives us this versatility, we propose to prioritize and maximize our design capacity and control over the interaction between each cell and its microenvironment as a good predictor of the expected phenotype. And where we can also design the trajectory of a cell within our systems and thus be able to allocate differential and sequential treatments in time in order to have a replicable process regardless of its scale. And in nature, we found the inspiration we needed to think about this solution. Coincidentally, laminar flow and microvascular systems are widely observed technologies in many organisms as means of maintenance and control of growth and development of cells and tissues. Based on this powerful insight, we wonder if using laminar flow and microvasculature as technologies, we can propose a continuous process of biomanufacturing. Today, we're going to tell you that story. 7 years ago, we asked ourselves for the first time if it was possible to design a platform that allowed us to implement laminar and continuous bioprocesses automatically, leveraging the benefits of the microvasculature that we find in nature. These 3 microfluidic devices are the heart of the bioprocessor. Their goal is to replace and automate the steps of scaling up a cell culture, from manipulating the cell bank to harvesting processed medium ready to be purified. The first device, strain-on-a-chip or cell-on-a-chip, consists of an arrangement of fluid traps that modulates the ratio of cell division and escape from the system, to provide a constant flow of available cells to initiate the scaling up process. It can be inoculated under a biosafety cabinet, then frozen or dehydrated for later being used within the production facility. This device eliminates the need to have a cell bank on site and the need to manipulate cell banks or reactivate them when we want to start a production process. The second device, a two-dimensional bioreactor on a chip, is designed to be able to monitor the behavior of the cell line during the first cell division cycles of the bioprocesses, and to calibrate the output cells density, the formulation of the media, the pH, the oxygen dissolve online. This device facilitates the calibration and optimization of parameters in real time. But perhaps the most distinctive and challenging of these three devices is what we call the bubble-free bioreactor. The objective of this device is to functionalize the efficiency of the microvasculature in a three-dimensional system that allows the correct mixing, oxygen dissolution, feeding, and pH and temperature control. The benefits that we have detected in the use of microchannels and laminar flow in the bioprocess are the removal of cellular stresses and the greater control over the microenvironment, which means that we are able to work with higher cell densities in some cases and having even greater efficiencies in the conversion of media into the target protein. In addition, it is a continuous bioprocess. So, in comparison to traditional ways of scaling up microorganisms that are in batch approaches. The characteristics of this device facilitates the reduction of the footprint and the install facilities to achieve the same processing volume, but between 100 and 400 times less footprint installed. Today, we're going to do a deep dive on each of these devices, reviewing our development processes, the challenges we encounter, what we have achieved, and what is to come. The bubble-free bioreactor is a three-dimensional microbioreactor printed with porous biomaterials. We call it microbioreactor because it uses microchannels to guide cells through the system, and not necessarily because of its size. This device facilitates the control of oxygenation, pH, mixing, and density control in a laminar regime without the use of any internal moving parts. These two independent channel systems shares an intermembrane, which facilitates the exchange of mass and energy. The contact surface between these two phases is comparable to that of the lungs, with more than 20,000 square meters per cubic meter. And continuing with this comparison, fluid with cells and in suspension and nutrients circulate in one of the phases, and gases to oxygenate the culture media circulate on the other one, with also controlling pH and temperature. When we intervene this lattice with a gas and liquid feeding system, we ensure that the microenvironment of the cell is kept constant as they move inside the bubble-free bioreactor. By manipulating the feeding rate, we can calibrate the speed of the cells and their residence time in the system, thus balancing the number of cell divisions, the uptake of culture medium, and the nominal cell density. To create this device, we turn to the study of lattices and crystallography. In this case, we are using a double gyro because it has two independent channel systems, and this minimum surface maximizes a very efficient way of increasing the contact surface between these two phases. In order to be able to design our microchannel structure, leveraging these crystals in large scale, we require a virtual suite to render such high complexity topologies. Once the design has been rendered, this virtually designed object is post-processed into images and fed into a 3D bioprinter that is capable of achieving the feature size resolution for microchannels manufacturing. The 3D printer will then project these images into the printing bath containing photocurable materials to actually build the final piece. For the intermembrane to be able to perform its function, we need its material to be gas porous and biocompatible. In order to build these devices using 3D printed, the first thing we needed to develop was a photocurable, porous, and biocompatible material that would provide the functionalities that we are looking for in the intermembrane. Do we have the biomaterial to manufacture the remaining 99% of the bioprocessing industry? Our biomaterials team explored a multi-parametric space for the development of a photocurable biocompatible resin with an internal porous structure that offered the possibility of gas permeation. The materials are designed also to provide drug delivery properties and promote or enhance natural cellular mechanisms. Also, we are taking into account the mechanical resistance that can provide the BFB with the strength and low mechanical erosion. To achieve these objectives, an extended group of biological and inorganic compounds had been tested to develop new hybrid materials with specific properties for different cell culture systems and final products desired. As we can see, there are several subcomponents that make up a biocompatible and photocurable composite. Beginning this discovery process meant thinking outside the box and testing endless combinations to find good candidate formulations for this application through a broad and smart screening which do not exist in the market. First, we had to study and test different protocols to effectively be able to solubilize all these components in an homogeneous and rheological appropriate system to achieve a stable formulation over time that could be used as an ink. For each subcomponent, specific kinds of solvents, mixing procedures, secondary reactives, and temperature control phenomenon reactions were among the things that we had to study and screen in search of a successful blend for a 3D printing resin. However, a stable blend does not necessarily mean that it is photocurable. Therefore, we had to carry out calibration curves for each one of these formulations, defining its ability to be light cure or not. It responds to different thresholds for photocuring, and its response curve to different times of exposure and different light outputs. To characterize the behavior of a resin and their development, a working curve analysis must be performed. The working curve is a function that relates the layer thickness that is built and the exposure time of the light engine and the potency of the light engines. With these curves, now we are able to see the performance of a photocurable biomaterial to define the suitability of this resin. And once we have detected these precandidates that are compatible with the photocuring process, we proceeded to evaluate their properties as printable composites. To guarantee the resistance to mechanical stress, porosity, structure, and biocompatibility of the biomaterial, different parameters were studied in order to homogenize the light curing process, as well to describe the arrangement of the microstructures and to manipulate the delivery of the non-reactive compounds after the printing process. After the cross-linking screening, most consistent biomaterial formulations were subjected as candidates to print cylindrical coupons for post-curing in-house mechanical stress testing. The 3D printing parameters for these coupons were selected according to working curves of the photocuring properties of each biocompatible material. To measure the resistance to mechanical stress, we perform mechanical tests such as tensile exposure and 3 bending flexure tests. With this test, we were able to relate the effect of the different components of the 3D printed biomaterial by knowing the correlation between the stress applied to the biomaterial and its strain. The characterization was the last stage to decide whether a hybrid material is a printable good candidate or must be discarded. The mechanical characterization was carried out on a pre-selection of formulations. Specimens were produced according to the standard and were analyzed by an external supplier whose methods are certified for tensile, bending, pressure and twisting tests on polymers. The results obtained from these traditional tests allow us to see the big picture and to visualize with great sensitivity the effects of each variable on the mechanical properties of pieces printed in functionalized biopolymers. We can see the format of the results and the variability for a subgroup of pre-candidates in this graph. We had to develop a specific platform to be able to measure the porosity of our biomaterials, which already had the necessary properties to be used for 3D printing process. We designed a device that allows us to diffuse culture medium and oxygen simultaneously, joined by an intermembrane with a known effective surface. This allows us to know the flow rates of culture media and gases and the measurements of change in pH of the culture media at the outlet. And by knowing this change in the pH, we have the possibility of calculating the quantity of infused gas into the culture per square centimeter of the known intermembrane of the device. In this image, we can see the effect of gas diffusion on the color of the culture media, indicating a change in pH due to the CO2 dissolution that diffuses through the membrane. Once we were able to certify that the pre-candidates qualified on all the listed properties, we began to study the interaction between these biomaterials and cell lines and cell cultures. In order to achieve this, we had to develop a post-curing, pre-washing and pre-incubation protocol to ensure the biocompatibility of the pieces. After the potential candidates were selected, we began to do the biocompatibility tests. To prepare the piece, a post-treatment, a washing treatment and a sterilization protocol were carried out to avoid any contaminations or biomaterial leaching during the test. Here we can see some of the results of these trials where we were able to measure safety for several of our candidates throughout cell culture. As we can observe, the cell population grows without apparent changes compared to conventional materials. And now, with this selection of biomaterials that meet all the requirements to build bio-free bioreactors, we have to carry out a calibration print to understand how the material behaves when used in the construction of large pieces. This allows us to create the digital signature of the biomaterial so the printer can incorporate the specific considerations when printing each type of material to be able to build these reactors successfully with all the design parameters well achieved and liquid tightness. The last test carried out was to measure the survival of the pieces throughout time. Stability, durability, fragility, resistance to internal pressure, to manipulation by operators, and to integration with new connectors were considered. We are proud to share with you that after running this biomaterial screening and optimization pipeline, we work with more than 100 interchangeable compounds and hundreds of candidate formulations. Today we have a library of more than 10 biomaterials qualified for this application. This gives us a lot of versatility in the design and application of future microfluidic systems. This work is the foundation of our ability to develop and optimize biomaterials. Our goal for the future is to functionalize them to carry out a specific task for each cell line. So, now that we understand how to formulate, manipulate, and test photogradable biomaterials, we are one step closer to being able to build microfluidic bioreactors. Microvascular systems contain a large volume of topological information. If we think that our circulatory system contains a total volume of 5.5 liters of blood, can you imagine the complexity of designing and visualizing the entire topology of the circulatory system in a virtual design suite? We have tried this for a long time, and what we learned from most of the software that we tested is that when the microvascular system exceeds a total internal volume of 20 milliliters, the suites crash, and these systems are impossible to manipulate. We quickly understood that in order to scale the microvascular systems designs that we were going to strive to get, we will have to design a suite specifically optimized for this application. So we asked the question, can we design the remaining 99% of the bioprocessing industry? When we reflect on this challenge, we return to nature and study the patterns and rules it uses to order such a complex problem with no apparent effort. Then comes the powerful insight of designing large-scale microfluidic systems, describing them as self-similar fractals that can be generated automatically by establishing simple low-level rules and allowing the system to grow in order to observe emergent behavior from the topology and thus iterate over our design cycle. Today, for the first time, we present Caster, our suite for large-scale microvascular system design. To start this process, we have to interpret space as a group of subunits that transition between different geometries. We realized we could use these subunits of space as Legos to build higher hierarchical structures. Caster allows us to choose from a library of geometries and bespokely change the parameters desired on the minimal subunit we want to use within a really easy and intuitive user interface. Once we have chosen the type of subunit that will occupy a specific space, we can proceed to embed the crystallographic parameters that we want to govern that subunit of space. This will be instrumental in defining the relationship between phases, the effective contact surface, and the microfluidic function of that space. Now that we have chosen the subunit and the crystal that we want, we can use them to build higher hierarchical topologies, ensuring the continuity of the microvasculature. We can now set several rules, even on those higher hierarchical structures, in terms of being able to establish growth relationships of a volume in order to balance the effect of cell division and cell density throughout the system. Once we were able to incorporate the high-level design parameters and we are happy with the topology obtained, we can proceed to functionalize the lattice so that we can incorporate culture media and gas feeding systems, as well as collection systems to unify the inputs and outputs of the microbioreactor. In real time, we can see how different low-level parameters emerge automatically, which allow us to obtain insights on the go, to choose the best option considering the function and the final volume that we want the microbioreactor to occupy. Now that we have a structure already functionalized as a microbioreactor, we are ready to print it. And Caster allows us to do the slicing and post-processing of the piece in real time and build the image library to be able to print it later or send the information to the printer as we print each layer of the piece. The caster allows us to visualize, design, and render large-scale microvascular systems on a laptop with a refresh rate of up to 400 frames rate per second. And the slicing and processing can be done by generating images of virtually any resolution. So when we say large, that can be a really vague term. So it's valid to ask, how large, right? Analyzing the context of this demo day, we thought, what would be big enough? Can we simulate a system with a volume equivalent to a human circulatory system? Maybe 20 circulatory system, maybe 1,000 liters. So we thought about making an attempt to simulate a microvascular system with a total volume of 16 million liters of total volume, equivalent to the total global installed production capacity for the biopharma industry. And this was the result. So nature inspired us. Being able to take advantage of its wisdom to design solutions fills us with the desire to see the challenges that we can conquer together. Once we have the biomaterials and we know how to arrange them, the next step is to have a machine that can effectively materialize a microbioreactor. We chose 3D printing as the best technology, taking into account the level of complexity and data required to build a large-scale microbioreactor. The big question is, again, can we 3D print the remaining 99% of the bioprocessing industry? In this graph, we can see which are the light-based 3D printing technologies currently available on the market, according to their surface and working resolution. On the one hand, we analyze the limiting factors of the options available in the market. We see that the technologies that can reach smaller pixel size are laser-driven, scanning laser 3D printing or two-photon polymerization. And on the other hand, when we look at the technologies that are optimized to maximize the working surface, they make use of high-resolution light projectors, capable of exposing a large number of pixels simultaneously, where the size of the image is amplified to the desired working surface. Laser-based photopolymerization has its limits. When working with a single point, the maximum speed of the laser is inversely proportional to the minimum dose of energy required by the material to be polymerized. Also, when working with many moving optical parts, the obtained precision reduces as the printing surface becomes larger. However, the number of pixels per layer can be infinite. On the other hand, for DLP, this approach has limitations too. The maximum resolution of the projector and the desired pixel size limits the 3D printing working surface. As you can imagine, we want to print large surfaces without compromising the size of the pixels. So we ask ourselves, can we manipulate each pixel of a projector as a laser is in a SLA printer? In order to do this, we need to have an array of several millions of micro lenses lined up in the same pattern as the pixels are in the projected image. Each of these micro lenses should have a specific design to be able to focus each one of these pixels in a very small movable area that can scan the whole pixel surface, increasing the maximum resolution. We then took on the challenge of designing this array and securing a partner to build it. In this photo, you can see a portion of this microlens array. Once we have the microlens array in our hands, we were able to test our hypothesis and observe that the effect that we were looking for existed. Now we had to incorporate this phenomenon into a 3D printing protocol. Today, for the first time, we are presenting our brick printing technology. Our brick printing technology leverages the benefits of printing millions of dots simultaneously with the precision and versatility of a laser-based printer. We have designed an optical system that allows us to work with highly collimated projections that are perfectly aligned with our array of microlenses, allowing each projection pixel to focus on a single point of light of variable diameter. On the other hand, we incorporated a piezoelectric device that allows us to move parts within our optical system with a precision of 20 nanometers in the X and Y axis, allowing us to scan the original pixel size area with the spot produced by the microlens array, thus increasing the final resolution of the projector perceived in the VAT. We call it brick printing because we manipulate each focused pixel within its influencing surface by light curing boxes that are arranged like bricks in a wall, completing the total volume. Once we were able to verify this phenomenon, we proceed to design an optical system that would allow us to build the first 3D printer with brick printing technology. We had to find a supplier company that could build the aspherical lenses we needed. Since its size and complexity made it very difficult to order, and not only we did find the right partner, but we were also able to move forward with the construction for the printer during the pandemic. Once we have all the parts, all the components in the lab, we were able to assemble the optical system and turn on this system for the first time. We decided to use a chess pattern to check that there were no crosstalk between neighbor pixels while we manipulated the XY position of the pixel. After the first adjustment, this was the result. Each of these dots corresponds to a pixel of the projected chessboard pattern. We have little moving dots. Why is this important? Being able to focus each pixel of light on the specific size spot that we want and manipulate them in a space allow us to increase the printing surface without losing pixel size, maintaining resolution while printing millions of points at the same time. Also, since the piezoelectric has a 20 nanometers precision, we are able to print features that are even smaller than the dot produced by the MLA. Today, for the first time, we show our new line of 3D printing systems with brick printing technology inside. Cambio. The first model of our Cambio systems is called Esclarate, and it features an effective printing volume of 29 liters, 84 teraboxes within that effective volume of printing, a print speed of up to 590 million pixels per second, 14 centimeters in the x-axis, 28 centimeters in the y-axis, 60 centimeters in the z-axis, with a pixel resolution of 6 micron and resolution per layer of up to 983 million pixels per layer. Our projection system is embedded with the firmware to process the input from the caster to print in real time and in synchrony with the suite. The printer tray is contained within a temperature-controlled sterile cabinet where we can formulate the biomaterial in real time and ensure its homogeneity throughout the long print. Once the printing process is finished, we can remove the pieces and their support, maintaining sterility. We then post-process the piece, seal, and irradiated, leaving it ready to be used. This technology allows us to take full advantage of the progress made so far in the projection industry in terms of building high-volume parts with microvasculature resolution. This new branch of the company will focus on continuing to push the boundaries of rig printing and on building partnerships with key different players of the industry, since the field of application of this technology is much broader than bioreactors. Here we can clearly see the manufacturing potential of Cambium, potentially unlocking the possibility of printing several tons of microbioreactors daily. The scale of this is difficult to gauge. For example, a 32K printer printing every day will be able to build an installed capacity of bioreactors comparable to the worldwide installed capacity for pharma. We have already installed our first S-Claret Cambium system in Buenos Aires, and we are ready to continue optimizing this technology. Throughout this year, we work on the optimization and scalability of the bubble-free bioreactors. First, we work on obtaining an optimal separation between the phases and on the interface of the connection with standard elements. Then, we work on gradually increase the internal volume and incorporate a built-in feeding system, as we can see in the video. Afterwards, we incorporated a second feeding system to start producing functional bubble-free bioreactors out of the printer. Today, for the first time, we're going to show you the first bubble-free bioreactor that we printed with a total volume of 1 liter. It was a great event that kept us awake for more than 20 hours. Here is the first 3D microbioreactor printed with photocurable and biocompatible material with an internal volume larger than 1 liter. The speed at which we are scaling our systems is vertiginous. During 2021, we want to surpass the lines of the 10 liters of total volume. Now we're going to briefly cover the progress that we've made on our other 2 microfluidic devices, the strainer on a chip and the bioreactor on a chip, and also the integration of these 3 microfluidic devices to perform and carry out continuous bioprocesses in laminar flow. We perform an iteration process on the development of these devices to optimize their ease of use and construction. In our laboratories, we have all the necessary equipment to develop, prototype, and scale the production of our microfluidic devices. The strainer on a chip is a device designed to allow the use of different biological platforms, cell lines, without handling equipment or maintenance of the cells on site. Once inoculated with the cell line, the device can be preserved in liquid nitrogen until it's necessary for starting a new bioprocess. By connecting the device to the bioprocessor, the thawing and the activation process begins, and the cells reactivate their growth. Then the chip begins to deliver cells in an ascending manner until it reaches its nominal production capacity. To understand and design the thawing, activation, and feeding process, we had to carry out numerous finite element simulations in order to make sure that the device was behaving properly. In this video, we can see the renewal rate of the culture medium within the traps and understand the optimal workflow to balance feeding, cell harvesting, and cell division cycle. This is a device designed with the purpose of studying cells behavior within the chip, in order to corroborate that the phenomena that we described in the simulation was actually happening inside the microfluidic chip. For each cell line, we carry out optimization tests where we observe the movement of cells within the chip and measure viability and the number of cells that escaped from the chip over time. In this video, we can see how cells flow without apparent effort in a stream of culture media. Depending on the speed of work, we can also use a flipping system to counteract the effects of gravity if the flow is too low. In this video, we can see the microfluidic effect of the traps inside our chip. They alternatively generate a high-speed flow, allowing part of the cell population to partially move, and also allow speed areas to prevent cells from completely escaping. With each new cell line we work with, we have to carry out calibration curves to detect the right working parameters to achieve a stable throughput of cells over time. The bioreactor on a chip consists of a series of feeding stages that allow us to control physical, chemical, and biological parameters throughout a certain number of cell divisions in order to synchronize the cell population at the chip exit. In this video, we can see the calibration of the feeding stages, in which we seek to regulate the feeding rate and mixing of homogeneity. The bioreactor on a chip has 3 feeding stages. Here we are testing the system with color liquids to illustrate the transit of the cell through it and the continuous feeding and diffusion that the device confers. The bioprocessor integrates the 3 devices, allowing us to control in real time the formulation of culture media and dissolved gases, pH, temperature, and cell density. It also allows us to treat specific parts of the bioprocesses in order to switch, for example, the metabolism. Furthermore, the device generates a bank of biological samples throughout the bioprocess. Finally, it allows partial harvest without the interruption of processes or the disruption of their sterile sealing. We have developed the operating units to provide these services independently, and last year we have begun to integrate them. Here we can see how some of the first integration prototypes that we designed were carrying out cell cultures by integrating our 3 microfluidic devices to optimize treatments in order to maximize antibody production in this case and the efficiency of use of the culture media. In this platform, we can carry out growth and production optimization tests using the different cell lines that we have in our laboratory, as well as industrial cell lines of our commercial partners, in order to carry out a first evaluation of productive metrics of the bioprocesses adapted to a laminar and continuous bioreactor. We are also carrying out the integration of all the electronic components of our hardware platform that provides the services described to our microfluidic devices. These prototypes show us a strategy to cancel sedimentation effects in the microfluidic channels, where we are able to measure parity between the entry and exit of cells from the system, showing that we were able to counter-arrest the effects of gravity and sedimentation. Perhaps more shocking is that in this space we have integrated all the steps from cell line activation, inoculum production, to scaling and harvesting of the grown culture media. With this, we close our tech review session of the demo day. Thank you for sticking with us through this journey. Hopefully, we found a good balance between the ground that we wanted to cover and the technical depth that we wanted to cover. Today, we spoke about the great challenge that we face regarding sourcing the materials we need sustainably, and how bioprocesses can play a transformative role aligning our relationship with nature. Also, the challenge that it represents to scale this industry 450 times. We presented our vision on what we need to change and where we can start building from. We presented the tools that we have developed to start building this future, the product that embodies it, and how we have started testing it for biopharmaceutical applications. On our technical review specifically, today we have covered our effort oriented to developing and building the tools necessary to physically carry out bioprocesses. STAM's ecosystem contemplates the integration of these tools within a highly regulated industry. So we are currently developing solutions focused on facilitating quality control online, the release of batches, and continuous optimization of protocols and cell lines. And we will cover this in these projects in future demo days. We are excited about the future of bioprocessing industry and how the bioprocessor can enable new roles for this technology. We want to start building the remaining 99% of the bioprocessing industry, but this journey has just begun. Join us. We certainly still have to face many challenges moving forward, and we will discover many opportunities as well. During the 2021, we will focus specifically on building commercial relationships with the industry, carrying out engineering reports, installing pipelines for integrating our technology into current infrastructure, and making our way into commercial scale with our current partners. Please reach out to us if you are curious about further details regarding our technologies and potential application cases that you would like to pursue. Also, if you have detected opportunities to work together, we would love to get in touch with you. As our team grows, we are constantly looking for passionate and high-skilled professionals and scientists willing to help us build our vision. We admire and acknowledge the great work being done in the bioprocessing industry, and we appreciate the experience of current players. We would love to engage with them in finding ways to accelerate our impact and capabilities to address this great challenge. All the work presented today is proprietary and protected by worldwide patents. Lastly, we want to acknowledge and recognize the daily efforts of the STAM team. We are really proud of the human group that we have built together. Their passion, talent, resilience inspire us every day and fuels our mission to develop laminar flow bioprocesses of excellence. Our offices in both Buenos Aires and San Francisco enable us to develop and engage every day with state-of-the-art technology in biotech, microfluidics, optics, biomaterials, electronics, IT. We want to thank you for taking the time out of your day to learn more about our work, and we hope that this event sparks your curiosity on how laminar flow bioprocesses can play a key role on tackling great challenges, and encourage you to engage with us so together we can explore action plans to build the remaining 99% of the bioprocessing industry. Thank you. Now we will have a few minutes to answer your questions.